Hi Terry, can you hear me?
Hey Sarah, can you hear me?
Yes.
Can you hear me?
Yes, can you hear me? Can you hear me?
Hello? Yeah, I can hear you now. Perfect.
Okay, good.
Hey, how are
Hi.
you? Happy New Year.
Happy New Year to you too, thank you. How are you?
I'm good, thanks. I'm good. Have you moved?
Yes.
Yeah? How's Montreal?
That's okay. Yesterday it started snowing, so I can feel the cold coming up.
Yeah? What makes you feel better? We've got a little bit of snow also here, but nothing too crazy. It's a little, so it's like in the lows, you know, like 2-3 degrees.
Yeah.
So that's okay. We'll get through it. So how you doing? You ready for next few days? I mean, when is it? Are we talking about Thursday, right?
Thursday, yeah.
Wow,
Hopefully.
Thursday it is. Okay. So let's go through the presentation. And can you follow up on a room available, like a room with Marianne?
Yes.
Or sorry, Anne-Marie. Anne-Marie. I'm not sure what she's done, but that has to happen today. And please CC me, because I'm just looking over here. Do I have anything? I just got a hold. I don't have a. Yeah, he needs to follow up with her. Okay.
Okay, sure. Should I also, you will be sending the email to other supervisors, not the committee members?
Yeah,
regarding
yeah. I can send an email to them.
Okay.
Or you can send it. Actually, I think it's fine.
Okay.
One thing we have to do is we're gonna have to do. I've been thinking about it. Are you gonna come back for this?
Yeah.
Okay. I think it's better that way. So we're gonna do a hybrid. So Jean Paul is gonna be in online, but the rest of the folks will be in person. Okay.
Okay.
So we need to get that room. So yeah, okay. So just find the room and then we'll figure out who can send the email.
All right.
And after you finish your presentation, please send me a copy of your presentation. I was hoping to have taken a look at it already so I could give you more feedback, but we can go through what you have right now. So let me make you, okay, you should be able to control the screen now.
Okay. Can you see the screen?
I can. Just give me one quick second while I do something. All right. Okay, I'm good.
Okay.
So you have 25 minutes, okay?
All right, so good morning everyone. My name is Sarah Hajeri and today I'm excited to present my thesis project, elucidating leukemic stem cell interactions with the tumor extracellular matrix niche. So acute myeloid leukemia, or AML, is a heterogeneous hematopoietic cancer. It's the most common acute myeloid leukemia in adults, and its incidence rate increases with age. It has poor prognosis in elderly with 20% survival rate at 5 years, and it's characterized by irregular expansion of undifferentiated myeloid blasts. The primary treatment involves chemotherapy and stem cell transplantation. Here you can see the abnormal myeloid cells in an AML patient. So sorry. There has been it has been suggested that mutation in genes regulating proliferation.
Sarah, can you do me a favor?
Yeah.
Sorry for interrupting you. Can you quickly add slide numbers?
It's over here.
Where?
There. Can you see?
Oh, there it is.
Yeah.
Thanks. Continue.
Okay. It's been suggested that mutations in genes regulating proliferation or differentiation in hematopoietic stem cells or progenitor cells can give rise to leukemic stem cells. And these leukemic stem cells have self-renewal potential. They can divide rapidly to sustain the growth of leukemic blastocysts. And and they're the reason behind the relapse of cancer patients as they're chemo-resistant and they can regrow the disease. Here you can see the niche of hematopoietic stem cells and leukemic stem cells. It contains of secreted factors, other cellular components, extracellular matrix, and other things. Knowing that these two cells, leukemic stem cells and hematopoietic stem cells, share the same microenvironment in the bone marrow, in myeloid malignancies like leukemia, studies suggest that leukemic stem cells can modify or remodel the hematopoietic stem cell niche into a leukemic niche. And this remodeling selectively targets the leukemic stem cells and suppress the normal function of hematopoietic stem cells. So it's really important to understand the interaction between cells and their ECM niche. So studying the leukemia niche is crucial for understanding the development, the progression of leukemia, and by investigating this niche, we can identify the cellular and molecular interactions that promote the leukemic transformation and find new targets for a more effective treatment. So here you can see the important cells, the receptors that are involved in the niche. So the current methods to study the cell-ECM interactions are summarized in this table. Xenograft models are the gold standard model. They're used to detect tumor samples in immunodeficient mice. The issue with them is that they're labor intensive. They're used to detect, they're technically challenging, and it doesn't give us the understanding of what ECM is. Then decellularized bone marrow. Bone matrix is another method that involves removing the cellular components from ECM, and it doesn't give us the exact ECM cell interactions. It just gives us an ECM source or backbone to seed the cells on them and study their behavior. Another important method is synthetic and natural scaffolds involving 2D, 3D, and co-culture systems. This system is really important to studying the cell-ECM interaction, and because it gives us the exact ECM components, and then cells can be cultured on them and we can see their behavior. So if we want to study the hematopoietic stem cells or leukemic stem cells in vitro, we can take the samples from patients and grow them in regular polystyrene dishes. Even with addition of cytokines and growth factors, they quickly die and lose their biological functions within hours to days. They die because they're lacking a niche in vitro. So I am hypothesizing that by using this approach of adding the cells to an effective culture system, we can actually culture the primary AML samples on these matrix constructs incorporated with ECM components, and the cells can regrow and continue to return their biological functions. This approach allows us to study the ways the cells interact with their niche and gain a better understanding of how these different components contribute to biological functions, cell survival, proliferation, and differentiation. So current understanding of hematopoietic stem cell niche is that it's consisting of two niches. Some cells can reside close to vascular niche and some close to the endosteal niche. And I selected hydroxyapatite to be a representative of an endosteal niche and elastin to be a representative of vascular niche. So to recap, I'm creating conditions that hopefully patient-derived cells can continue to grow and return their biological functions in it. My first objective was to define the critical ECM components for leukemic stem cell survival, differentiation, proliferation using scaffolds efficacy assay. So PBMNCs, peripheral blood mononuclear cells, will be incubated on different ECM matrix constructs for 72 hours. We do viability assessment using Trifon Blue. We do functionality assessment using CFU assay, and we do phenotypic changes assessment using flow cytometry. Then I'm going to, the second aim was comparing leukemic and healthy niche to understand underlying niche mechanisms using molecular interrogations. We did differential gene expression analysis using bioinformatics study. The first plan was to use RT-qPCR using TaqMan assay, but we decided to hold on it. Then we proceeded to select a druggable target protein using bioinformatics studies. Our second aim was to utilize the selected ECM to conduct a drug screening experiment to identify agents that selectively target leukemic stem cells. So we did in silico screening of 1,280 biological active compounds for optimal docking to a selected protein. So for my first objective, I made 2D scaffolds. I fabricated this. Collagen type 1 flakes are mixed with acetic acid to rehydrate them overnight. It's then blended and degassed to make a homogenized slurry after addition of different concentrations of elastin hydroxyapatite. 50 microliters of each combination is then dispensed in 96 cell plates, and plates are left open in the fume hood to allow the liquid to evaporate overnight. So for the scaffold efficacy assay, we taught 5 patient samples in total. We seeded 50,000 cells per valve for each scaffold combination. After 72 hours of incubation, we recovered the cells from the scaffolds, and then the cells that are recovered are then mixed with medicold for the CFU assay. After 14 days of incubation, the number of CFUs were counted under the microscope. We also did CFU assay on AML patients before the incubation to act as a positive control. Additionally, the cells in colonies in each valve were also pelleted down and stored for the protein or RNA analysis in the future. So for the results, here's the CFU readout results. For each ECM scaffold. This is the results normalized to T0. As you can see, although the results of our study didn't yield a statistically significant finding, it's worth noting that there was an observable increase of the size of the bar graph. It's more than 1.5 increase in mean of number of CFUs compared to T0. Because of the heterogeneous nature of AML cells, this increase may not reach the threshold of significance, but it suggests a potential trend or pattern that can be further explored. And I wanted to pick the best ECM condition, so I normalized the result to polystyrene here. And ultimately, we chose conditions collagen, hydroxyapatite, elastin 1, 0.3, 0.3 because it had the highest statistical significance. Then I choose 0.7, 0.3 because it yielded the highest mean number of CFUs, and 0.3, 0.7 because both high mean and also statistically significant. And this finding is considered a significant breakthrough in this field because it suggests there's potential for population expansion and maintenance of multipotency of these primary cells. Here is the representative colony image. So we can see CFU-GM, CFU-E, CFU-G, and CFU-M. So I wanted to gain deeper insights into the impact of system on AML cells' behavior. So we scaled up the model of the scaffolds. We created them in 6-well plates to accommodate a larger volume of cells. Each well was seeded with 3 million cells. And after recovering the cells, we used trypan blue for viability assessment. We stain the cells with different antibodies to see the phenotypic changes, and then we did functionality assessment with CFU assay. So here you can see the viability assessment results in patient 3. Here you see that we seeded 1 million cells, but we recovered more than 3 times the amount that we started with. Knowing that the process of recovery involves 3 rounds of washing, and then you're losing some cells in the process of lifting the cells from scaffolds, it's a considerable breakthrough that in these 2 patients we see in some conditions you're getting expansions of cells more than the initial seeding concentration. Then here you can see the percentage of live cells, and again in one of our conditions you can see that with polystyrene less than 50% of cells are alive, but with this condition almost 70% of the cells are alive. So even though we were getting large number of cells, this doesn't define the progenitor potential. So we're not interested in getting simply the expansion of bulk leukemic blast cells. So we needed to do CFU assay to look at the colonal units. Here are the CFU readouts for each ECM condition. You can see that we had a significant increase in the number of mean CFUs compared to control in our experiment, and we did 3 functional and 3 biological replicates, so N is N of 9. Then we wanted to see if we can detect the phenotypic changes when we seed the cells on the scaffolds. This is the flow cytometry results. CD33 plus 45 plus is the myeloid leukemic cell marker, and noting that these patients samples have leukemia and it's really heterogeneous. It's based on the genetic and even the age of the patient. So this results, when we wanted to compare them, you can't really compare together. It's better to see the results within patients. So the cell frequency of antibody conjugated AML cells at day 0 was compared to 72-hour seeding on different ECM conditions. So here we can confirm that the readout is leukemia. We're not getting expansions of lymphoid cells or something unusual. Then this is CD33 45 plus, DRD2 negative, and again we see that compared to T0 we're getting the expansions of the cells. So these cells have potential for these scaffolds have potential for population expansion and maintenance of multipotency. So for our next objective, we wanted to initially do a bioinformatics study to validate the concept. A microarray dataset was obtained from GEO database. Differential expression analysis was performed on 19 healthy control CD33 plus peripheral blood sample and 10 peripheral blood leukemic samples. And we did a Lima package with bioconductor, and here are the genes that are upregulated and downregulated. I should say that I selected the genes that are related to ECM. If you see collagen type 5 alpha chain 1 is downregulated, and important upregulated genes in AML are integrin subunit alpha M, integrin subunit beta 2, CD44, integrin subunit alpha X, and other important ECM factors. I also did pathway enrichment analysis. I inputted the genes that were changed significantly in the system and the pathways that are enriched, ranked by combined score. This combined score is a score that combines important statistical factors like p-value and t-value. Here we can see interleukin-2 signaling pathway and monocyte and its surface molecule pathway have really high combined scores, and important ECM genes that are involved in these pathways are also highlighted. And again, this is a representative of these two important pathways. Recent studies show that in leukemic model, inflammation is an inflammatory pathways are really important. So here you can see most of the pathways are inflammatory, but monocyte and its surface molecules are also really important. So for my next objective, I wanted to utilize the selected ECM to conduct a drug screening experiment to identify agents that selectively target leukemic stem cells, and I did in silico screening of the compounds for optimal docking to selected protein. So I selected, I wanted to find a target gene or protein to do my in silico studies on, and then we can validate them on with experimental design. So the selected target protein was integrin alpha M beta 2 active domain, because I saw that in my differential expression analysis, both of these genes are significantly increased in expression in AML samples. So here in green, you can see the integrin alpha M. Here it's integrin beta 2. This is commonly known as CD11B, CD18, or MAC1 protein. And studies show that expression of this gene is associated with poor prognosis in AML patients. This is the methods for in silico screening. So prior to virtual screening, the proteins and ligands needed to be prepared. This involved addition of hydrogen bonds, Kalman charges, and preparing the right format. Can you see it changing? Okay. So Topris Mini 2 drug library was used. It contained 1,280 biologically active compounds. It was the library obtained from the company was in SDF format. So then the library needed to be processed into individual drug molecules, and these individual drug molecules was then transformed from 2D format to 3D structures, and then we used Autodoc Venus software to do the virtual screening. The software engaged all 1,280 drugs in docking process with the integrin alpha and beta 2 structure. So here you can see after the docking simulations, the results containing critical information such as PubChem ID, the name of the drug, and the most important thing, respective binding scores. So the lower the binding score, the better the binding. And here we selected the 5 best drugs and we did ligand protein. We showed the ligand protein interactions. Here in the background, you see the alpha and beta 2 integrin ectodomain, and this is compound 628. And you see alanine amino acid 443 and lysine amino acid 252 are most important amino acids that are repeated for binding of our compounds to integrin alpha and beta 2 ectodomain. Here's the next. This is the third one, the fourth compound. So as you see, there are different compounds that went into different grooves of integrin alpha and beta 2 ectodomain. So to summarize, this is the ligand receptor adhesion pairs involved in hematopoietic stem cell and leukemic stem cell retention in bone marrow niches. You see how the bone marrow niche is modified, different interactions occur, and studying of these interactions are really important. And ex vivo studies are crucial to explore the molecular mechanism involved in leukemia. For future plan, I suggest that we can use a blocking antibody against integrin alpha and beta 2 to analyze the effects on peripheral blood mononuclear cells. We can repeat the scaffold screening with CD45 plus 33 plus cells to only analyze changes in myeloid cells and not the whole peripheral blood mononuclear cells. We can optimize the scaffold like addition of other ECM components like hyaluronic acid and going from 2D to 3D scaffolds. We can perform RNA-seq and validate with RT-qPCR plates. We can perform drug screening on the most optimal scaffold. Here you can see the Tachman array human extracellular matrix and adhesion molecules. It contains 92 target genes associated with ECM. So this is our next step that we're going to analyze the cell ECM interactions. I wanted to acknowledge our group members, NSERC funding, CIHR funding, and other collaborators, and the biology department. Thank you so much for listening, and I can open the discussion.
Hey Sarah, sorry, my mouse is a little bit weird. Okay. You were perfect for time. All right, perfect. Let's just go step by step through stuff. So I'm glad that you. So first of all, you have a spelling error over here. Collaborate Mark Minden. It's D-E-N. And maybe you can put a photo of the group.
Okay.
And can you get the CHR and the insert logos, if you can insert them?
Mm-hmm.
Okay. Can I put you in just a quick hold for 1 second? It's about the radiator, okay?
Sure.
Hey, Sarah, can you hear me?
Yes.
Perfect. Sorry about that. It's about the radiator, so I just got to make sure. Okay, so I think you did great. I really do. I think you're good. Like I would say like 95% there. I just have a few suggestions that maybe we can go through from the beginning. So your timing is great. Your tone right now, to me, is I'm sensing I'm kind of tired, I'm bored, I want to get this over with, I need to graduate. Which is fine, it's normal, but you need to infuse a little bit more enthusiasm, a little bit more like excitement, because it helps to show that a little bit of passion, okay? And it's because you're talking to me and it's on Zoom, but when you're doing it live, excitement, like energy. You need to show energy, okay? Because that really helps to convince the people. And this is one of the reasons why you're doing, going in live, that you're excited about this, and we gotta infuse that excitement within your data too, so they understand what are the real cool things that you've done, right? And so I'm hoping we can go over some of the cool stuff, right? So, getting to your slides is, can you share it again, or can you do a full screen share, right? Okay. Maybe you should, oh, that's why. Okay, perfect. Okay. So this is great. Next slide. Okay, this is good. I'm going to ask you, and this is the type of question I would ask. So here you mentioned the primary treatment is chemotherapy and stem cell transplantation. Is it an autologous or an allogenic stem cell transplant? And can you explain why?
So it's ablation of native bone marrow and then transplantation of, I think allogenic. I have to double check on that because it's from another healthy donor to the patient.
Okay, it is allogenic, but why is it allogenic? Why do they use an allogenic transplant and not an autologous transplant?
Because we need the healthy stem cells, right?
Yeah, but an autologous transplant also has healthy stem cells.
But it's in a deformed or malignant niche.
So, okay.
So the cells are extracted from not healthy donor, as I'm guessing.
Okay, so, but what if you could infuse completely healthy hematopoietic cells, autologous? And let me, just to save time. So the reason why they do an allogenic transplant, is actually twofold. One is we can't actually purify hematopoietic cells and leukemic stem cells. So if you can't purify them, you can't separate them out, there's a good chance of reinfusing leukemic stem cells back into the patient, okay? That is not the case for myeloma. In myeloma, they can actually do an autologous transplant, right? Because the nature of the disease, the CD34 positive cells or hematopoietic stem cell, or it's the hematopoietic stem cell fraction is in that fraction, versus AML, which has the same CD34, right? So one of the reasons. The second reason for an allogenic transplant is that the allogenic immune cells that get generated actually help to combat if there is any residual disease. Okay?
Okay.
But what's the biggest risk with an allogenic transplant?
The antibodies that are not native to the patient, and then the inflammation and the risk of...
What's that condition called?
Oh, I don't know.
Graft versus host.
Okay, yeah.
So please read up, okay, on an allogenic and graft versus host disease, because they may ask you this. Okay? So graft versus host is really bad.
Yeah, it might reject different
really bad.
things, yeah.
Okay? And so it's you're saving the patient with an allogenic, but at the same time, there is a big risk of graft versus host if it goes, it's basically your immune system goes on steroids.
Yeah.
Because then the new immune system goes on steroids. So that's one of the complicating factors, okay? So I think it's important that you just read up a little bit about the background, because it may be a question. I got to ask you something, all right? Okay. So I'm giving you a heads up. Okay, next slide.
This slide is fine. I don't need to add anything or combine.
No, I think it's fine. This slide, my only issue here is I think the font needs to be bigger, like hypoxia, secreted factors, growth factors. It's a lot of empty space, and you need slightly bigger font because I can't read it.
Okay. For only the diagram.
The diagrams.
Okay.
The diagrams. All right. Here, you need to point out vascular niche and endosteal niche. You need to point it out over here.
Okay.
Okay, like you need to literally put a circle, vascular niche, right? Put another circle, endosteal niche. You need to highlight that the vascular niche is close to your endothelial cells. Your endosteal niche is close to your osteoblasts. Okay.
Okay.
And this is part of this is the current understanding of where these hematopoietic cells, and by extension, leukemic cells, are somewhere between, right? There's still some controversy, but this is where it is. So you need to identify vascular and endosteal over here.
Okay.
Okay, good. Next. I think this was fine.
I don't need to change the font.
No, I think this is fine. Okay, so here we talk about hydroxyapatite and elastin. You almost want to have that same diagram on the side to remind people when we talk about endosteal, what we're trying to recreate is the ECM of this component, the elastin one, right? Because that involves the vascular niche. Okay? And hydroxyapatite is a part of the endosteal, right? Because what we're trying to recreate is the ECM of the endosteal. Okay?
Okay.
So I think you have to put in context. You need to explain those two niches, and you need to explain how you're adding these components in an effort to recreate, part of the ECM of those niche components. Okay?
Got it.
So it's a little bit more than just I'm adding everything together. Like you've added these things because these are major ECM components, but the kind of the educated guess is that you've that the hydroxyapatite is representative of endosteal and vascular is representative of vascular, and you're creating different combinations which include single components and combined components too. Okay.
And we should talk about collagen as well.
Yeah, collagen is the glue that holds it all together. And remind people, it's the major protein in the ECM. Okay?
Okay.
How many different collagens are out there?
Oh, I have to read up on that. 12?
You do. Please. Last time I checked, there was 26.
Okay.
And what collagen are we using?
Type 1.
And where's collagen type 1?
Bone marrow?
In the bone. Well, it's
in
bones.
Bone.
It's in bones. So type 1 collagen is the predominant collagen in your bones. Yeah. Where else is it?
I don't know.
Sarah, do some background reading, okay? So things like it's predominantly connective tissue. So anything to do with your tendons, your ligaments. It's also in your skin. Type 1 and type 3 is in skin. But it's very common. So read up a little bit about it, right? Just because they may ask you, why didn't you use another collagen, right?
Okay.
And then I'm going to ask you, what is the source of your collagen?
It's a bovine source.
Okay, from where?
Company?
Yeah, I know. Which part of the cow?
Oh, femur, I guess. No?
No,
Tendon?
it's Achilles.
Okay.
The Achilles tendon.
Mm-hmm.
So it's type 1 from the Achilles tendon. And why are you using a cow collagen and not human collagen?
Ethical. It's more ethical. Ethics.
Yeah, okay. So sourcing, it's very hard to source human collagen, right? And is collagen evolutionary conserved between cows and humans?
Yeah, it is.
Yes. Do you know how much?
No.
Can you read up?
Okay.
Right? So I think it's like between 93 to 97% evolutionary conserved, like amino acid sequence identical. And why do you need an identical amino acid sequence?
Because we ultimately want to recreate the niche of humans. So.
Okay, but it's a little bit more nuanced than that. So collagen, what can you go through primary, secondary, tertiary, quaternary structure?
So.
How does it fold?
I have to check on that. Alpha and beta sheets, and then.
Beta sheets.
Okay, so alpha sheets.
So what is the tertiary structure of collagen?
Oh, the fibrils you're talking about?
Yeah.
Okay.
I'm looking at it right now.
The meshwork of the three fibrils that are interconnected together.
Yes, it's called a triple helix.
Okay.
All right. You have 3 alpha helices. You have an alpha 1, two alpha 1s, and an alpha 2. Right. So you have amino acid sequences for for an alpha 1 and an alpha 2. 2 alpha 1s and 1 alpha 2, right, make a triple helix. Okay. And it's a very special way of winding up because you actually have glycine on every third amino acid residue. Because you have glycine, it allows the triple helix folding.
Okay.
Right? If you didn't have it between cows and humans, you wouldn't have a triple helix. So that's why it's evolutionary conserved, which is why we can use animal ECM to represent human ECM. Okay? Important to understand.
Okay.
Okay. Next slide.
So I have a question. When we fabricate our scaffold, when we do acetic acid and then form a 2D film, how are we sure that these helixes are maintained?
We are sure. I'm just going to put you on hold before I answer, okay? Hey, Sarah, you still there?
Yes.
Sorry, I'm just dealing with elevator. I thought my life was going to be easy, but it's not. Just let me quickly send this. Okay. So your question was, how do we know it doesn't denature, basically, right?
Yes.
We've been, for various reasons, the acetic acid and all these conditions that we've done, during my PhD, I validated that there's no denaturing. So it works really well. There's no denaturing so what you have when you actually swell it in acetic acid is you do have an unfolding of the triple helix, and it swells it, right? But then when you precipitate it, okay, so when you freeze it, you're actually precipitating it again. It refolds into its triple helix. Okay? And we've known this because we've done things like DSC and all that stuff that measure what is ultimately, it's considered melting. So collagen as a biopolymer, right? And this is what polymers do. When they start to unwind, that is a melting effect. And you can measure their melting by using a differential scanning calorimeter, okay? Because that tells you when the molecules have started to unfold, basically. And so we've done it, okay? So it's all good. But good question. Okay, I'm fine with this. Yeah, this one, I think what you needed to do is you probably may want to, instead of having like 2 click-throughs, just put it on from the beginning. Do you know what I mean? So if you go back to the previous slide. Yeah, so slide 7. So keep it like this from the beginning. Don't have a click-through. Okay? Next one.
And is it fine that I did RT-qPCR and I...
No. Don't cross it out. It just asks a question that you don't need someone to be asking. Okay?
So I just don't need to talk about it at all.
I would just remove it completely from there. If it comes up, you'll talk about it. Okay? But you're focusing on your differential bioinformatics study. Okay?
Okay.
Perfect. Okay, so we're slide 20 now. Hang on, we skipped ahead.
Oh, yeah.
Go back, all the way back. Skip ahead. Okay, so...
Here.
Go.
This is also fine?
Yeah.
Okay.
Hang on, sorry. Go back. Go back. Get back to where we are. Paste. Okay, so that's fine. Okay, so these are all fine. Perfect. Okay, next. Okay, this is good. I have no issues with this. This is slide number... Okay, slide number 11. You need to explain what CFU is. You have to. Okay? Nobody knows what the method called CFU assay is. Nobody. You can explain colony forming unit. You need to explain it's a semi-solid where you suspend the cells in methacellulose, which is spiked with differentiation growth factors and cytokines, okay? Such as stem cell factor, interleukin, you can look them up, right? EPO. So it is a condition that allows you to suspend cells within a 3D semi-solid, right? At a fixed number of cells, and actually then count the number of colony-forming units. And because of the different cytokines, it focuses on the differentiation in the myeloid lineage. So you can detect granulocytes, you can detect monocytes, right? You can detect erythrocytes, right? And so you can get your composition of this multi-lineage differentiation, right? And it gives you a frequency, because now you know the number of cells. It's only a single cell that gives rise to the colony, and you detect it over 14 days. Okay?
Okay.
The same conditions can be used to assess a patient who has leukemia. But because of the leukemia and the enhanced leukemic burden, what you now generate are these what are called blast colony-forming units. Okay? So these blast cells are these tumor cells. Because not every single leukemic cell gives rise to a blast unit, right? So it's a functional assay of measuring the number of progenitors in a patient sample.
Okay.
You have to explain it here.
Just a quick question. So the lymphoid line, it doesn't have any progenitors? So that
It can't read out the lymphoid because we don't have, so the Methocult doesn't have the cytokines, and even if it did have the cytokines, the lymphoid lineage is very difficult to differentiate in vitro.
Okay.
That's the issue here, okay?
Okay.
So, this one, all right, you need to say it's an N of 3 biological repeats. You also need to mention at the very bottom somewhere, just write, what is your stat test? What did you use?
Okay. ANOVA.
You did ANOVA, right?
Yeah. Okay.
Yes?
Yes.
That's an ANOVA?
ANOVA,
Okay.
yeah. One
All
way.
right. Perfect. So you just please put that down. Secondly, so the left and the right, let me understand, what's the difference between the left and the right? Is it the same graph? Is it the same data?
This is normalized to T0. This is normalized to polystyrene.
Okay. Perfect. So that's important. I think it's important to understand. But you need to explain it a little bit better because it looks like it's different data. So in the first set of data, you're normalizing to T0, okay? And what and can I just ask, is there any significance between your polystyrene and your T0?
Yes.
So why don't you have that?
Yeah, I have to. Okay, maybe. Okay, I have to check again because I checked that as well. If I don't put any, I don't think that there's any significance.
Can you please just check?
Yeah.
Because I have a feeling that your polystyrene, your HA, your elastin, your collagen, all those are what's it called? Significantly reduced.
Okay.
Right?
Oh, so I just only put the ones that are significantly.
Yeah, I know, but it's not only those. You put those because you've now normalized to your polystyrene.
Okay.
Perfect. Okay? But I almost wonder if you shouldn't even put A in. Don't put A in. You don't need to. Just keep it at B. Okay. You're just complicating the message. Right? The only thing I would do is, you know how you have your polystyrene? You should say polystyrene 3 days. Like T 3 days.
Okay.
You need to explain that this is polystyrene at 3 days and T0 is the time of thaw. Okay, so that's why you're using polystyrene as you're normalizing, right?
Okay.
Graph number A, can you put it as a separate graph and keep it at the end of your slides, like at the end, right? So if someone asks you that question, you have it, you can pull it out of the bag.
All right.
Okay?
Okay.
So this is slide 12. Okay, and we said you're going to measure biological. You're going to say biological, right?
Yes.
Okay. Next. I think this is all right, but you need to label them.
Okay.
You need to tell what people are, right? And I always wonder if you should just, when you describe, I think this slide, when I said you had that flow chart of the schematic of what you're doing, like the next slide could be, these are the CFUs that are being generated. Or how about this? Is there a way, so if you take slide 13, go back 2 slides. Those representative images, you can put them, let's say, on the right side. So when you're explaining the CFU assay, this is what they look like.
Okay.
All right? And then what you do is you receive the composition and the total number, all right? Because what you're showing is the total number of CFUs.
Yes.
Not composition, right? So I think you basically get the CFU stuff out of the way. And, these are the types of representative colonies that we make. Okay?
Okay.
So put in that slide. So this slide 13, just put it in 11. Okay?
Okay.
So then afterwards, so you have data, then you have this. This is your scale-up method. All right, so you've talked about CFU now, functional and phenotypic, okay. But the reason why you're scaling up is you move now from your 90, you didn't make that clear. You moved from a 96 wall, now that you identify, I don't know if you should call it scale up. Because, or maybe you just need to explain it. So you've identified the matrix that you showed. Now you're going to scale it up in 6 wells, and you're going to test how these cells perform in a 6 well, and then measure them through functional and flow cytometry. So it's okay, keep scale up. But just explain a little bit better. Okay?
Okay.
Okay, next. So this is 3 separate patient samples. You need to explain to folks, these are 3 patient samples, right? These are patients who, they donated samples. The likelihood these patients have also passed on now. All right? This is the reality. They've passed on. And you're measuring trypan blue, and what you found is you found a couple of samples where you had an increase, right? So if you, I guess, the percent live cell, this is percentage.
This
and
is after
an
3
M3,
days?
Right? And this is cell count.
Yes.
Okay. So what you're saying is you're generally getting more cells coming out. You're right. You can see that in between your samples, you were getting more live cells and more trypan blue cells, right? One thing that you need to change, Sarah, you see on the bottom right, and I think you had this on the previous slide. Go back to your previous slide. So you see the schematic you have where it says viable cell and apoptotic cell? Does trypan blue measure apoptosis?
No.
No. What does it measure?
The metabolism marker?
No. That's methylene Alamar blue.
Oh.
What does trypan blue measure? Read up.
Okay.
Trypan blue it's a necrosis marker. Apoptosis is measured through a bunch of other things. Ultimately, an apoptotic cell will transition to a necrotic cell. But an apoptotic cell in the early stages, right, has its cell membrane intact. A necrotic cell is when the cell membrane is starting to dissolve, break up, okay? And trypan blue measures necrosis.
Okay.
And you have a bunch of biology people in there, so change that.
Okay.
Okay? So you measured cell number, okay? And even though you're getting lots of cells, all right, it's important to mention that these cells do not necessarily discriminate between what are the functional blast-forming units or what are just leukemic cells, all right? And in order to make this distinction, you then repeated your CFU assay, okay? Which is what you've done here. And this is really exciting stuff, right? Because here, you see again, you have your normalization to your, this is your time of thaw, right?
What do you mean time of thaw?
You've normalized the time of thaw.
To polystyrene.
Well, if it's normalized to polystyrene control, polystyrene would be at 1.
Yes, it's, T0.
Just T0, okay? So, in your statistical significance, is there any significance between your T0 and polystyrene control?
No.
Okay. But generally speaking, you see the similar trend as there is a reduction in blast-forming units, blast CFUs, okay?
Okay.
But what you did see, right, in 2 conditions, you were able, in 3 different biological samples, right, you were able, and this is how you need to make things exciting. 3 different patient samples, right, there is a tremendous amount of biological variability between 3 different, these are not cell lines. These are 3 patients, okay? You found that you actually were able to get a significance Are you sure the significance was done to your T0 or is it done to your polystyrene?
Yeah, it's done to T0.
Okay. Again, you put the test in there, but you were able to show that you had that. This is extremely exciting because no one has been able to keep these cells alive long enough in a dish. And now you have a condition that can keep them alive enough in a condition in 3 separate patient samples just by playing with the ECM. Okay? You've got to make it exciting. This is a freaking breakthrough. Okay?
Okay.
16. Oh, can you go back one slide? So you see the red line? Can you please put T equals 0 at the end?
Okay.
Because you need to explain what the red line is. Right? And then I think the same thing goes for the next one, right? Are you going to put a red line or no? No, don't put a red line. Don't put a red line, okay?
Okay.
Okay. Okay. But you've explained why now you've gone to the CFU method, because now it's not about the number of cells, but it's actually the number of blast-forming cells. Okay?
Okay.
Perfect. Good. And then you decide to go into the phenotypic analysis. So what does CD33 measure?
It's a myeloid marker.
Okay. What does CD45 measure?
Hematopoietic marker. That's a...
Not true.
No.
No, it's not hematopoietic.
Oh, so hematopoietic except RBC and lymphoids.
So it's pan-leukocyte. It's leukocytes. Okay? Because you see, what's Q4 over here in your flow cytometry? What is that?
Cells that are 33 negative, 45 negative.
So what cells are they?
Maybe platelets, RBCs, leukocytes.
So, not leukocytes. Where would leukocytes be?
Oh, just 45 plus here.
Yes. So Q3 and Q2 are leukocytes. Q4 are 45 negative. So 45 negative means it's probably RBCs. Platelets are too small. All right, so you probably have a percentage of RBC contamination in there. Okay?
Okay.
So what's Q3?
So these are 45 positive cells.
And?
Q3?
Q3, yeah.
33 negative.
What does that mean?
It's not myeloid cells.
So what is it then? Not myeloid, but leukocyte. What is it?
I don't know.
What are the lineages of your white blood cells? Myeloid and
Lymphocyte.
Lymphocytes. So those are lymphocytes. Okay, 45 positive, 33 negative. Okay, they probably, in order for you to hand on heart say that they are lymphocytes, you would have to stain them with like CD19. Okay? But in all probability, they are lymphocytes. Okay? Which then goes up to your myeloid cells. So 33 and 45, it's a double positive. So it's myeloid and panleukocyte. Okay? So that's why you can say that Q2 is of the myeloid, and because there's such a high percentage of them, and you know that it's from a leukemic patient, this is the leukemic component. All right, these are mostly leukemic cells. Right?
Okay.
And what you've done is you've looked at these leukemic cells. You've purified, FACS allows you to look at the percentage of these leukemic cells. Right? So what you're showing me is, what are you showing me? What are you showing me in this graph?
Just to compare the percent of CD33 positive, 45 positive between the 3 components that we had.
Yeah, and so you're not getting any significance, right? So whatever effect you're getting, all right, you're not changing the frequency. Well, you kind of are changing the frequency a little bit because of graph patient one you are,
Yes.
right? So you are changing the frequency a little bit, but it seems to be generally consistent between
All.
polystyrene and the other conditions, right? So when you look at polystyrene as your variable, you're not changing the frequency, right? So whatever changes have happened, right, it's consistent between the ECM and the polystyrene. But what this is telling you is that you're still enriching for a blast-forming unit, even though you've maintained a consistent frequency. Do you see what I mean? Does that make sense?
Yeah, but what about patient 3? It should be
not significant between polystyrene and... So patient 3, that's the one that you have the highest amount, isn't it? Of blast-forming units.
Well, it decreased from T0 to my condition.
Yeah, I know, but go to your CFU data for patient 3. So this is Tripen Blue data.
Oh, I didn't put the CFU data for different patients.
Okay, you pulled it together. Okay. Well, I think what I'm saying is, generally, so even though you've got, so yes, there's been a reduction. So, and the conditions are ultimately, I guess, you know, you're losing a percentage of your cells for sure, of your leukemic blast cells. But whatever's remaining there seems to be enriching. Okay? Because if it was just a chronic reduction, you would have seen whatever the Tripen Blue number was telling you, right? You would see, you should have seen less CFUs. But the fact that the percentage is staying the same and you're getting more CFU, it's probably telling you getting an enrichment. Okay?
Yeah.
All right. This is number 17. Okay, good. Next. Okay, this is the combined data?
Combined data, yeah.
Can you go back?
So
why wouldn't you show the combined data and the flow cytometry plot?
What do you, yeah, like I should put this here or?
Yeah, so if you're combining all the data and you're normalizing it, right? It's all normalized, I'm assuming.
Yes. So this is 33 plus, 35 plus. This is DRD2 negative as well. So I'm excluding the stem cell fraction here.
Well, but you know what? Don't talk about DRD2. If it's about DRD2, I wouldn't talk about it. You haven't focused on it at all.
Yeah.
Right? So I would actually put this graph at the back somewhere.
Okay.
Stick to the individual ones, okay?
Okay.
Yeah, it's just gonna complicate your message. You need simplicity.
Okay.
Yeah.
All right.
This is good. No issue here. Go next. Okay. All right. This is fine. And you're highlighting that you're going for alpha M beta 2 because they are upregulated. Okay?
Yes.
So this is what you need to focus on. You're showing that you have found in a subset of, 19 healthy versus peripheral, that there is an upregulation of alpha M beta 2. At this point, right? The next slide after this is you need to introduce what integrins are.
Okay. So
you need to say integrins, right? Are the molecular connections between the ECM and, they are transmembrane heterodimers where they come in alpha chains and beta chains, okay? And when they dimerize together, they create an integrin, and then these integrin cluster to make a focal adhesion. You need your next slide has to be a schematic explaining integrins.
Okay.
Alpha and beta. And just remind everyone, you're focusing on the alpha M beta 2. And the alpha M beta 2 is believed to bind to what? What does it bind to?
TIMP and so it has promiscuous binding to different things, but TIMP one is one of the important ones.
List everything it's supposed to bind to.
Okay.
Okay? Mm-hmm. Because I'm assuming that it binds to some kind of ECM component.
Yeah.
Yeah? Mm-hmm. And that's the story here, Sarah. The story is you're figuring out that there's an integrin that is upregulated. We're now adding an ECM in here, right? And it allows what you're hoping is happening is that the ECM is allowing a proper connection with the alpha M beta 2, which is why we're getting survival. That's our working hypothesis here. Mm-hmm. So you need to connect these things. You can't just throw, integrins. Because Nima is in there. Nima doesn't know anything about integrins. Mm-hmm. So explain it to him. Okay? But, so alpha and beta 2.
Mm-hmm. But this results, it's not cultured on any scaffolds. And,
I know. Okay. You're just showing that there's an upregulation based on public databases.
Okay.
On 19 healthy versus 10 patient samples. Okay? Mm-hmm. And you use that to then do your in silico mining.
Yes.
Right? This is fine. You need to explain what the red's for.
Mm-hmm. So these are only the ECM factors in the pathway. So these are all the genes that were upregulated or downregulated. Combined, the score is really high. And in this pathway, these are the ones related to ECM. So.
Yeah, so this is our subset of ECM genes, right? That we curated from ultimately the TACMan array card, right?
Yes.
Okay, next.
It's the same thing.
Okay. Yeah, that's fine. And then you do that's fine, everything's good here.
So here, the reason I'm focusing here, because the alpha and beta 2 is also a monocytic marker. Should I talk about this or forget about it? Because then it's monocytes and then it's AML.
No, I'm fine. I think it's good. It adds to the story. Monocyte is part of the myeloid lineage. Mm-hmm. Okay?
Okay.
Next. Okay, that's fine. So I think where you lost people is you now have a target. Mm-hmm. Okay, the alpha M beta 2, which you came across because you found that ECM has some components. So you're really focusing on ECM. Mm-hmm. And your main target is your alpha M beta 2. So you're actually now doing an in silico screen to identify something that could potentially be a druggable target on this membrane-bound receptor. Mm-hmm. Okay? And that's what you're doing. You're going through an in silico to find something that binds to the alpha M beta 2. Mm-hmm. Because you're hoping that is an anti-leukemic agent. Mm-hmm. Right?
Okay.
So, you've gone through this. That's fine. That's all good.
So that's what I should explain, and then I'll go through.
Yes. Mm-hmm. Explain. You're trying to figure out an anti-leukemia agent now. Mm-hmm. Okay? You're doing all this so you can figure out an anti-leukemia agent.
Okay.
Okay?
Mm-hmm.
Alpha and beta 2.
Okay.
All right.
Mm-hmm.
Next.
And this is okay.
I was fine with this. Mm-hmm. Yeah, that's all good.
This one?
Yeah. So you're highlighting a compound, right? This compound over here that you're saying, compound 628, with this unpronounceable long name. That is a potential hit, right?
Mm-hmm. So I focused on 5 first hits.
Yes, this is the first one, second one, third one. Okay. So you've now created a short list of potential candidates that need to be tested. And if you had time in the future, you would test them. Mm-hmm. Okay? But this slide is a great slide to end with, because where did you get the slide from?
Oh, I should add the reference. It's one of the...
You need to add the reference, okay? Mm-hmm. Where's alpha M beta 2?
Here.
Where?
In the leukemic stem cell here. Alpha and beta 2.
Perfect. Can you put a little... You're focusing on this one. Mm-hmm. Can you put a box over that?
Okay.
So you're focusing on that as a potential anti-leukemic target. All right?
Okay.
And all of this is being discovered because you've been working with an ECM that is showing that you can keep things alive happier. So what if you can now target that and actually kill the leukemic cell? Those are the terms you need to focus on, okay?
Okay.
It's not only about keeping them alive, but now you have a druggable target. Okay?
Okay.
Okay, that's all I have. And I think you're going to be good.
Okay.
All right? And we talked about this. I'm going to put you in a quick hold because I need to send a quick email, Sarah.
Okay.
But I think you're good. Are those comments all right?
Yes. So is there any way we can have another meeting just before 11 so that everything is...
Yeah, there is. Yeah. But can you update what we said? Send it to me?
Yeah.
Okay. And then you need to find out the room booking, please. Ask for something in LSB, obviously.
Okay, sure.
Sorry, I got to deal with some TAs. I have to go to Burj now. Okay, I think you're good.
Okay. When should be our next meeting?
Send me the thing. We'll lock in the date soon, okay?
Okay, all right.
All right? But update what you have.
Okay, thank you.
And practice. Please practice.
Okay. Just like a silly question. You talked about once with black attire, it shows something. Should I wear black or color?
No, you should wear something that's nice and formal. Don't come with track pants, okay? Something nice and formal. I'm not saying black or anything, but formal.
Okay.
Like a formal thing. Bring some coffees. Bring some little desserts or something, right? I don't know. Bring like a Persian dessert or something, right? Just for people to just break the ice a bit. And do you want to send the folks in the lab a note because everyone can come and join us for the presentation? So please have them join us. Okay? So that's on you.
All right.
Okay? We're good?
Yeah, you're good. Thank you.
Thanks, Sarah. Okay, bye-bye.
Have a good one. بهتون دادم